This is going to be part 3 of our discussion of chapter 3 of our Human Anatomy and Physiology text. So far we've talked about the cell membrane, aspects of membrane protein function, the cytoskeleton, and a number of passive membrane transport functions. Now we're going to take a look at active transport in contrast. So active transport moves solutes across the membrane against their concentration gradient using the energy from ATP molecules. Just to review, passive membrane transport refers to processes by which solutes move across the membrane with their concentration gradient, meaning from high concentration to low concentration. Active transport moves solutes the opposite direction, from low concentration to high concentration. That's what we mean by against the concentration gradient or difference. So cells have to invest ATP energy in order to make this happen. So what we're going to do is take a look at each of these different examples of active transport and see what's going on. Primary active transport is the movement of ions across the membrane, or typically ions across the membrane against their concentration gradient using ATP energy. So we're talking about a carrier protein, kind of like what we talked about with passive facilitated diffusion, but in this case it's not a passive transporter, it's a transport protein that can hydrolyze ATP. So it's an ATPase enzyme, we say, which then allows it to transport a species against its concentration gradient. The example we're going to use of primary active transport is called the sodium-potassium pump. All your cells have on their membrane sodium-potassium pumps that transport sodium ions out of the cell constantly, day in and day out, potassium ions into the cell using ATP. They do it cycle by cycle, pumping those ions with an ATP and starting again, pumping ions with ATP. So cycle after cycle. The net result is that there are concentration gradients, steep concentration gradients, we often say, for these ions across the membrane. What we mean is there's a large difference in concentration from inside to outside. So you can imagine if the sodium pumps are pumping sodium out all day, there's a low sodium concentration inside of cells, and that's what we see. The pumps are pumping potassium ions, K, into the cell all day, and in fact there's a much higher potassium concentration inside cells than outside. So that's the role of the primary active transporter. Here's a picture of various stages of the primary active transport cycle. Every time the transporter goes through a shape change, it's driven by ATP hydrolysis, and then at the end of the cycle, it's back to where it started, and another cycle transpires. So that's why this is drawn kind of in a circle. It's not going in a circle, it's just the same transporter going through different stages of its function, its cyclical function. So what happens? We see here, if we start at the top of this diagram, we have the transporter facing the inside of the cell membrane, and sodium ions can bind to specific binding sites inside the transporter. When all three binding sites just happen to capture a sodium ion just by random movement of the sodium ions, then this transporter can hydrolyze ATP, in other words, break off one of the phosphates of this adenosine triphosphate, and when that happens, energy is released. And that energy will drive a change in the shape of this transporter, its conformation, we say, and it will point the other way, and the sodium ions will diffuse away from the transporter. There is no longer a sort of a strength of attachment of those ions. They're kind of shucked out of the transporter. Well, once that happens, potassium ions located outside the cell will eventually, by random movement, bind to their binding sites within this same transporter that now become revealed when it's in this outer facing position. If you look back, you'll recall there are no binding sites for potassium when the transporter is facing inward. the protein changes shape, and one of the consequences is that now there are some specific potassium ion binding sites. So potassium ions bind to the transporter, and once they do, the transporter just automatically resets to its original position, and the potassium ions can diffuse away from the transporter. In addition, that third phosphate, that inorganic phosphate, that's what we call it, why it has that little I there, can diffuse away also. Inorganic means it doesn't have any carbons and hydrogens and oxygens like an organic compound. It's just a phosphate ion, a phosphate molecule, and some oxygens bound to it. All right, so it diffuses away, and now we're back to the start of our process, and then the cycle will repeat. Sodium ions will bind to the inside, ATP will be hydrolyzed, change conformation, release the sodiums, capture potassiums, and bring them in. So that's what the sodium-potassium pump does all day long, and creates gradients. So outside, this is just another way of illustrating the exact same thing, only in one picture. They just kind of shorthanded it, so just randomly. But it's the same thing. We mean to represent the sodium-potassium pump right here, and there's lots and lots of sodium outside the cell because we're pumping it out there all day, and the kidneys kind of make sure that there's plenty of it out there all the time. Not much sodium inside here, inside the cell cytoplasm. Well, there's another type of primary active transport that depends on the gradients that this transporter creates. It's called secondary active transport. There's another type of carrier protein that takes advantage of that high concentration of sodium outside of the cells to capture sodium ions along with some desired solute that's outside the cell. And when those two species bind to this membrane transporter, it changes conformation and allows them to diffuse away inside the cell. If you look at it on its own, it looks like a facilitated diffusion transporter that just happens to take two things at once, say a sodium ion and a glucose molecule, or a sodium ion and an amino acid. But it's a secondary active transporter because it utilizes the concentration gradient or difference created by the primary active transport to bring solutes into the cell against their concentration gradient. Or sometimes they bring solutes out of the cell against their concentration gradient. But let's focus on this example here. This particular membrane protein has a binding site for sodium ions and for glucose molecules. And unfortunately, the artist didn't take the time to draw the cyclical nature of this transporter in operation. So let's just imagine it facing out. And a sodium ion binds to it right here on this little rounded pocket. A glucose molecule binds to it on this little hexagonal pocket. And when that happens, it turns inward and releases those two back into the cytoplasm. As soon as those species diffuse away, it then switches back to its exterior facing conformation. And that happens over and over. As long as there are sodium ions and glucose molecules, this transporter will continue to capture the glucose molecules and drag them into the cell, even if they're becoming more and more scarce out here. The instant a glucose molecule binds, the transporter switches them to the inside of the cell. And the reason that can continue to happen is because it can never go back the other way typically, right? Because there's hardly any sodiums in here to bind to the transporter on the inside. There's bazillions of them out here. So because the sodium-potassium pump keeps a lot of sodiums out here with the help of the kidneys, but not very many inside the cell, this secondary active transporter can capture all of the glucose molecules that are found outside the cell. Or if it was a different protein, all of the amino acids of a particular type that are outside the cell. So this is a secondary active transporter. It does not hydrolyze ATP, yet it relies on the concentration difference of sodium to capture glucose and amino acids and other substances against their concentration gradients into the cell. So that's primary and secondary active transport. Very, very important processes in the intestine, for example, where we want to capture all of the organic nutrients that are in our diet. We want all the glucose molecules that we've consumed to be absorbed into the body, into these epithelial cells that line the GI tract. We want to capture all of the amino acids from the proteins we have digested and capture them and transport them into the cells that line the digestive tract and ultimately into the blood. So that's where it's very important to have this secondary active transport mechanism taking place. Also in the kidneys. We said the kidneys work by filtration of a large volume of water in small solutes into the capsule of the renal corpuscles, and then reabsorption of 99% of all that water and solutes as the filtrate, it's called, percolates through the convoluted tubules of the kidneys, the kidney tubules. We're using secondary active transport to reabsorb all of the glucose and all of the amino acids that squeezed out of those capillaries by filtration so that we don't waste them. All right, just a couple of other active transport processes that we should probably define so that if they come up later in our curriculum, we'll know what we're talking, what it means, what it's talking about. Endocytosis is the capture using ATP of substances or particles or materials from outside the cell to the inside. And exocytosis is the transport of vesicle-bound materials from inside to outside of the cell. So let's take a quick look at an example of endocytosis. In this case, some substance desired by the cell is being transported into the cell as a vesicle forms around some of the extracellular fluid containing this desired substance until it's completely enclosed in a vesicle, which is now broken off from the cell membrane and transported into the cell, probably along a microtubule. So now we have a vesicle full of some material that the cell wanted inside the cell. So that's called endocytosis, a particular type of endocytosis. We're capturing some molecules that we wanted from outside the cell. Typically, those molecules may be destined to be released on the opposite side of the cell, or they may be substances that the cell will retain for its own purposes. It may digest those substances by combining this vesicle, this endocytic vesicle, with a digestive lysosome, a vesicle that the cell produces full of enzymes that can break things down, in which case we could digest all the substances that have been entered into and recycle them or use them in a partially digested form. So the point is, we're bringing some substances into the cell by forming a special type of vesicle. This is an ATP-dependent process. It's a very complex process. ATP doesn't appear in this picture, but I did want to emphasize for you, it takes a lot of ATP for this whole process to take place. Phagocytosis, instead of just capturing molecules that the cell wants, solutes, phagocytosis is the capture of particles, solid materials from outside to inside of the cell by formation of a vesicle. So the cell must have receptors to recognize the particle of interest. It probably has some proteins on its surface that those receptors can bind to, and the cell knows, therefore, what it's dealing with. There's specific interaction between the receptors and the particle, and the receptors continue to attach around the particle, forming this vesicle, this amoeba-like engulfing of the particle until eventually it's entirely enclosed and brought into the cell inside of a vesicle, only this time the vesicle is called a phagosome instead of an endocytic vesicle, because this type of endocytosis is called phagocytosis. This is how macrophages, for example, can gobble up bacteria that get into our body when we injure our skin and then destroy them. Phagocytosis. Pinocytosis is a process by which cells, especially cells, epithelial cells called endothelial cells that line the blood vessel or that form the wall of blood capillaries, the smallest blood vessels, capture small volumes of liquid from outside the cell, from the extracellular fluid, and transport them across the cell and release them on the other side, for reasons that still remain somewhat controversial or not well understood. But yet there's this process of pinocytosis by which these cells gather microscopic droplets of solution and transport them across the cell. Pinocytosis. Receptor-mediated endocytosis. In this case, not only is the cell just grabbing some solute that it wants into the cell, it's grabbing the solute by recognizing it through binding of specific receptors. So this is probably the predominant way by which endocytosis actually occurs. So anyway, solutes, the cell otherwise would not know that the solutes were present. It binds to receptors, and the binding of the solute to its specific receptor is recognized by the cell, interestingly. The cell knows this is happening, and it starts to develop this clathrin-coated vesicle. These little purple things are proteins called clathrin, or protein complexes, and so the cell will then form this vesicle, which is brought into the cell. So now we have captured the solute of interest by virtue of recognizing it using receptors. Again, an active transport process by which the cell can capture these substances. The last thing we'll quickly talk about is exocytosis. When cells produce products that they want to deliver to the outside realm, environment, they do so by a process called often exocytosis. Not always. So here this cell has produced a product represented by the red balls. It's probably a hormone or an enzyme or something of that nature, something the cell wants to deliver to the outside. And so the Golgi apparatus will store this product in a vesicle and imbue that vesicle with some specialized proteins called snare proteins. And the snare proteins allow the vesicle, as it's just moving around in the cell, to bind, be captured by snare proteins in the cell membrane, and at the appropriate time, when the appropriate signal is realized by the cell, something that tells it, yes, now is the time to release your product, then the vesicle will fuse with the plasma membrane, and the contents will be released into the cytoplasm. So we synthesize the product, store it in vesicles, place the snare proteins in the vesicles, deliver them to the cell membrane. They bind to snare proteins in the membrane. Then, when the moment comes when, say, some calcium ions enter the cell, which is a signal, the vesicle will fuse with the plasma membrane and release its product, its contents, to the outside. That's called exocytosis, I'm sorry. So here's a nice electron micrograph showing a cell releasing some product that it's produced into the extracellular fluid by exocytosis. A very beautiful micrograph indeed. 100,000 times magnification. Electron microscopy is quite an amazing thing, that we can see things as small as these molecules that have been synthesized inside the cell that are now being released to the outside. We will not, there's a part of your chapter talking about electrical properties of cells and how the same concentration gradient for potassium and sodium across the cell membrane produced by the sodium-potassium pump is actually used to create an electric charge on the membrane of a cell like a little battery. But we won't talk about that now. It's here. You can take a look and see what you think of it. We're going to talk about that when we talk about Chapter 9. We'll talk about membrane potentials, or electric charges. So this concludes our discussion of excerpts of cell biology from Chapter 3 of our Anatomy and Physiology text. So join me next time as we begin discussing Chapter 4, in which we'll introduce ourselves to all of the tissues, the different tissue types that make up all the organs and organ systems in our body. Once we know what all the different tissues are, what they're comprised of, what their properties are, then we can understand the anatomy of all the structures that they're used to create. So that's the end of our story for today.